Welcome to today's lesson on the evidence for evolution, including antibiotic resistance in bacteria. In this session, we will explore how the phenomenon of antibiotic resistance serves as a powerful piece of evidence for the theory of evolution, illustrating the principles of natural selection in real time. In our lesson today, we will begin by conducting an in-depth analysis of antibiotic resistance in bacteria. We will then look at the challenges that this poses, before going into more detail about how antibiotic resistance is evidence for evolution. So far, you have had lessons about genetic variation, evolution, and natural selection. You've learned how genetic differences and environmental factors can influence the survival and reproduction of organisms, leading over time to adaptations in populations. This essential background prepares us to understand the specific mechanisms through which bacteria can develop resistance to antibiotics. Understanding antibiotic resistance in bacteria as evidence for evolution shows how evolutionary principles are not just theoretical concepts, but observable occurrences that have significant impacts in the real world. This insight is crucial for developing effective strategies to combat bacterial infections and managing the use of antibiotics in medicine and agriculture. Let's get started. To understand antibiotic resistance, we first need to recognize the role of mutations in bacteria. Like all organisms, bacteria can undergo random mutations in their DNA. These mutations are essentially changes in the genetic code of the bacteria, and they can occur spontaneously during DNA replication. Sometimes, these mutations can result in new characteristics, such as changes in cell wall structure, or the production of certain enzymes, which might reduce the bacterium's susceptibility to antibiotics. These changes can hinder the effectiveness of antibiotics, leading to antibiotic resistance. Firstly, mutation occurs. Within a bacterial population, random mutations arise. Some of these mutations may confer resistance to a specific antibiotic. When an antibiotic is used, it kills the non-resistant bacteria in the population. However, those with resistance mutations survive. The resistant bacteria now have less competition for resources and can reproduce more rapidly. They pass on their resistance genes to their offspring. Finally, over time, the proportion of antibiotic-resistant bacteria in the population increases, making the antibiotics less effective. These bacteria reproduce via binary fission, a process where one cell divides into two identical cells. Under optimal conditions, some bacteria can divide every 20 minutes. This rapid reproduction rate means that resistant strains can proliferate in a short period. For bacteria, antibiotic resistance is a significant survival advantage. When antibiotics are present, resistant bacteria can survive and continue to infect the host. This survival leads to an increase in the population size of resistant strains, posing major challenges. Firstly is the increased difficulty in treatment. Infected individuals have fewer effective treatment options, leading to longer and more severe infections. Secondly, spread of infection. Resistant bacteria can spread more easily within and between populations, contributing to widespread outbreaks. A major challenge is the emergence of superbugs. Superbugs are strains of bacteria resistant to multiple antibiotics. MRSA, methicillin-resistant Staphylococcus aureus, is a notorious example that often affects hospital patients and can be fatal if it enters the bloodstream. Overusing and misusing antibiotics have exacerbated the issue. Here are some examples. Prescribing antibiotics for mild infections or viral illnesses has unnecessarily exposed bacteria to antibiotics, promoting resistance. Another case of misuse is patients not completing their full prescribed antibiotic course can leave behind surviving bacteria that mutate and develop resistance. Additionally, in some countries, antibiotics are used in agriculture to promote growth and prevent illness in livestock. This practice can lead to the development of resistant bacteria, which can then be transmitted to humans through meat consumption. These practices have contributed to a significant rise in antibiotic-resistant bacteria. To combat antibiotic resistance, several strategies are crucial. Firstly, responsible use is important. Antibiotics must only be prescribed when necessary, and patients must complete their entire course. Secondly, regulation in agriculture. This involves restricting the use of antibiotics in livestock to essential purposes only. Finally, continued development and research into new antibiotics are essential to keep up with evolving bacterial strains, although this is a slow and costly process. The theory of evolution by natural selection is exemplified by antibiotic resistance. Antibiotics act as a selection pressure. Only bacteria with resistance mutations survive antibiotic treatment. These resistant bacteria also display natural selection, as when they reproduce, they pass on their resistance genes. Over time, the bacterial population evolves to become predominantly resistant. It is important to note that random mutations occur in bacteria, whether they are exposed to an antibiotic or not. It is not the antibiotic that is causing the bacteria to mutate resistance. Instead, it is the fact that any bacteria that have randomly mutated to be resistant to an antibiotic, the selection pressure, have an advantage and are more likely to survive and reproduce in the presence of that antibiotic. This process is a clear demonstration of natural selection, underscoring evolutionary principles. In today's lesson, we explored evidence for evolution, focusing on antibiotic resistance in bacteria. Evolution, driven by natural selection, involves species gradually changing over time. Darwin's theory explains how better adapted organisms survive, reproduce, and pass on advantageous traits. We examined how bacteria demonstrate evolution in action, particularly through antibiotic resistance. Bacteria with resistance mutations survive antibiotic exposure and reproduce, leading to a population of resistant bacteria. Real-world examples like MRSA highlight this issue in medicine. Understanding antibiotic resistance as evidence of evolution emphasizes the importance of responsible antibiotic use and ongoing research to combat resistant infections, linking evolutionary biology to human health.